Bonjour, we are the Puichi Macau team. This year, we are looking to support the global food supply chain and improve hydroponic farming using engineered bacteria, HTPS, hormone targeting and pH adjusting systems. Our project aims to improve current hydroponic systems with hormone binding domains and a new pH shooting system. To explain what we have done, there will be 4 parts to this presentation: project and problem and project design, experiments and results, hardware and modeling, and integrated human practice and outreach. We will now talk about some of the problems the global food supply chain is facing.
And these are problems we're facing that may disrupt the global food supply chain. Climate change and acute food shortages in developing areas around the world. Our globalized food supply chain is facing more challenges than ever. And to be part of the solution, our team is investigating the localization of food production, particularly in urban areas around the world. Our cities require large areas of farmland from across the world to supply their food demands. This means a disruption in this complex supply chain will severely endanger the food security of the urban population. As we can see from this graph published by the UN earlier this year, hiking food prices poses a real threat to the developing world. A possible solution is hydroponics, as it is vertically stackable and compact compared to traditional farming methods, and plants grow quicker in a controlled environment. However, hydroponics is not perfect. It costs more than traditional soil farming, and it takes a lot of man-hours to water and maintain. It is also not efficient enough to replace traditional farming to supply cities around the world. In our project, we aim to solve some of these hurdles in hydroponics and make it a part of the solution to our global food supply crisis. I'm now going to talk to you about our product design and our solution to one proposed. We plan to improve the current hydroponic system in 2 areas: efficiency and self-regulating level. To increase efficiency, we are targeting the germination phase. With our current technologies, the germination of seeds usually takes around 2 to 3 weeks. This timing is controlled and slowed down by plant hormones like ABA. We plan on using hormone-binding proteins, HPPS, to saturate and neutralize the effects of these hormones and reduce the germination time to around half of what it takes now. And to increase the self-regulating level, we're implementing a self-regulating growth medium that would eliminate the need for 24/7 manual and mechanical monitoring and intervention. The pH shooting circuits in our ECOLI will be able to react to pH levels in the growth medium and release safe and suitable amounts of basic acid or base chemicals. This would neutralize the growth environment and allow the plant to grow optimally. This also means our proposal has partially eliminated many man-hours in monitoring and repairing mechanical valves, sensors, and systems present with current hydroponics. To give proof of the efficacy of our project proposal, we will now talk about our experiments.
Now I'm going to introduce to you our experiment and results. So firstly, we transform GFP, green fluorescent proteins, into an E. coli BO21 strain with a PT11A vector. To investigate bacterial growth in different solutions, our team has established an E. coli strain transformed with GFP. We demonstrated that the E. coli grows significantly well in hydroponic solution compared to pure water solution in fluorescence rates and OD absorbance. Next, to monitor the pH level, the pH shooting system is composed of the base shooting circuits, PATP2 and LDHA. It produces lactic acid in the high pH environment. Also, as you can see here, we have another construct, the acid shooting circuits, PASR and GLSA, which is inspired by the BIT team of China in 2015, and it produces ammonia in a low pH environment. As you can see that the GLSA expressed in pH 5 in the Western blot experiments, and there's a high mRNA expression in both pH 5 and pH 6, indicates that the PSSR promoter is activated in acidic environments. Then we tested PASR-GLSA as it functions in a low pH environment. The pH change is very significant in pH 5 that the pH converges to 7 for the first 3 hours. Also validate that the SFGFP as our control, and also and our PASR-GLSA show low fluorescence in high pH environments. To sum up, GLSA proteins and mRNA expresses in pH 5, and functioned to neutralize an acidic environment. Also, it has shown a lower fluorescence rate in pH 9. Then, to improve PASRGL assay, we designed a pH shooting system with an acid shooting circuit and base shooting circuit, as we mentioned. The pH change of the genetic pH shooting system is larger than the control group, and it demonstrated that both acid and base shooting circuits functioned to neutralize pH 5 and 9. Next, the OD of the genetic pH shooting system is higher than the PASRGL assay in both pH 5 and pH 7. Particularly, as you can see on the right-hand side, the OD curve of the pH shooting system is higher than the PASRGL assay in pH 9, indicating that the base shooting circuits facilitated the E. coli growth in alkaline environments. Moreover, there is a clear band in the Western blot experiment of the pH shooting system at 30 kDa. As predicted, it is clear that the GL assay in the pH shooting system in pH 5 expresses the best. Overall, GL assay expresses in pH 5 and the pH shooting system functioned to neutralize acidic and also alkaline environments. It also has a better E. coli growth curve in the long term with its self-balancing system.
The constructs above were designed with PET 11A factors, which had a T7 system. To make a comparison, we improved our constructs with PSB1C3 factors, which have 2 biobricks. It is an optimal choice for our genetic pH shooting system, which consists of 2 circuits, each with independent promoters. In both LB medium and hydroponic nutrient solution, the results show that the newly designed constructs can function as well as the previous constructs, so they are a viable option for the implementation of our transformed E. coli in plant growth. We designed 2 constructs of hormone-binding proteins, PYL8, which is hypersensitive in binding ABA and inhibiting it. T2R4 is another protein that also binds to ABA with less sensitivity. Therefore, we added NSP4, a secretion peptide, to increase the secretion of the protein. In our experiments for hormone-binding proteins, we grew 2 groups of soybeans, one with added ABA and one without. Then, we added either PYL8, NSP4T2R4, or PET11A transformed E. coli to each group of soybeans. We observed the germination situation of the soybeans periodically and recorded the following data. As seen by the blue and gray lines, by the 4th day, the germination rate of soybeans with PYL8 was double that of soybeans without added cells or ABA. This demonstrated that PYL8 can discharge the effect of ABA and benefits the germination squarely. However, the function of NSP4T2R4 is similar to that of the PET11A control factor, so its function is not as outstanding. In our Western blot experiments, there were clear bands of 35 kDa for PYL8. However, the band of NSP4T2R4 is less visible, which may be why NSP4T2R4 is not efficient in inhibiting ABA. In the real-time qPCR test, RNA expression is present in both genes, indicating that protein expression is the problem with the T2R4 gene instead of RNA expression. In conclusion, PYL8 is significantly effective in inhibiting ABA, while NSP4T2R4 is less so. Both of them have mRNA expression, but only PYL8 has protein expression.
Now, let me introduce what we have done in terms of hardware in this project. Due to COVID-19, the global food supply chain is now facing food shortages. Hydroponics seems to be a promising solution. However, the costs are high. At first, a sizable hydroponic machine was used as it contains refined hardware that fulfills our needs. However, it produced a lot of waste and it was very space occupying. In order to tackle with these problems, we use a beaker to investigate if using fewer solutions will eliminate plant seeding. In our result, using the hydroponic machine required 5L more water and 0.5L more nutrient solution than the beaker required. Overall, using the beaker as a planting vessel would be a more cost-effective way and produce less wastes. We used wooden stakes to stabilize the plant in the beaker at first, yet it was not user-friendly and had to put most flatly. So we designed a cover using a 3D printer. We have to deal with the thickness, size, and amount of holes for the air pump and other sensors. Moreover, we applied in our PSB1C3 plasmid pH monitoring system. We managed to integrate the beaker, the cover, and the lighting in order to make it easier to promote it as a hydroponic home kit. The prototype was made with a laser cutter, and the components were combined with acrylic adhesive and ABS epoxy for the linking points to make it water resistant. The home kit is in a hexagon shape so that it can be mounted and arranged in a honeycomb pattern in order to better fit in the small balconies of Macau.
To support the hypothesis and design of our hydroponics improvement projects, we have modeled the yield of different common plants in various acid-based environments. We have also completed a brief quantitative analysis of carbon reduction if our project is implemented in our home city, Macau. In the first model, we analyzed 7 common food crops. We modeled each crop at 5 different pH levels from pH 4.5 to 7.5 and used Miller polynomials function and estimated 95% prediction intervals to fit the data. Here are the graphs of our results. We can see clearly that most of the major crops have a better yield in the pH 6 to 7 environments. This shows the importance of maintaining a neutral pH in our hydroponic system. In the second model, we have calculated how much carbon emissions from imported vegetables could be reduced by our project. We use data from Macau Institution and academic research on carbon emissions in supply chains to estimate the carbon emission. Figure 1 shows that with 10% utilization, we may procedure about 46 tons of vegetables, which is about 15 tons more than Macau's demand. Figure 2 shows that with 10% utilization, we can reduce carbon emissions by more than 12 tons compared to those produced by imported vegetables. It shows that at 10% utilization in Macau's amounts its citizens, then carbon emissions per year could drop by 44%.
Collaboration is essential to us. Amongst our various efforts, there are 2 that I would like to point out. Our first event was Sustainability on a Plate SOAP. It is related to SDG 2, 11, and 12. We invited iGEM teams and the public to prepare dishes with crops growing in eco-friendly methods and included a survey about hydroponics. It was shown that hydroponics is well recognized but uncommon. We concluded that people can incorporate green produce into their daily diets. Secondly, we organized a global webinar about aquaponics and hydroponics with the University of Macau's iGEM team. We attracted participating teams from Mexico, Macau, United States, and China. We exchanged valuable insights as we are all working in similar areas of interest.
The outreach department of our team is responsible for gaining insights from experts in different fields to constantly refine our projects while promoting our values to the local community. We visited Macau Urban Farm in Kowan in March to know more about the current situation of hydroponics in our city. We also discussed implementation processes with our long-term supporter, Win Hotel. We shared our project to Win and got to know more about food procurement processes of larger corporates. To study other branches of water-based agriculture in Macau, we also visited the Institute for Tourism Studies. For community services, we organized a workshop in which we guided students to make terrariums and taught them the basics of synthetic biology. We hosted in-school events like assembly talks in which we promoted iGEM and encouraged people to engage in solving global issues. We also participated in climate change conference in Macau to exchange ideas on environmental protection with leaders in the field. We hope to use what we have now and in return share them to the public. We have also engaged in integrated human practices to validate our projects and look for suggestions. At the very beginning of our project, we designed our 3 constructs, GLSA, the genetic pea shooting systems, and SFGFP using a PET11 effector. To compare efficiency, the staff from University of Macau Lab also advised us to put our constructs into the PSB1C3 system, which has 2 barrel bricks and is a better fit to our 2-circuit pea shooting system. We invited Professor Lam Hon-ming from the Chinese University of Hong Kong, who is an expert in designing and implementing hydroponic systems with the Hong Kong government, to share his agricultural research and give his opinions on our project. He gave suggestions on how to achieve food safety in our system. To familiarize ourselves with the food safety regulations in Macau, we visited the Laboratory Division of Municipal Affairs Bureau, the Macau regulator on food safety. We also learned about food safety tests and received support from the scientists.
Finally, in conclusion, we are using genetic pH in the system and homobinding proteins to enhance the existing hydroponic system. We're promoting self-farming and aiming to raise awareness for sustainable food production. Our system, HTPAS, is offering an optimized solution for the global food supply crisis we face today. HTPAS leads to sustainable food success.